This is an overview of an article entitled Genetic Variants Influencing Plasma Renin Activity in Hypertensive Patients from the Pharmacogenomic Evaluation of Antihypertensive Responses Study, published in the April 2018 issue of Circulation, Genomic and Precision Medicine. Hypertension is a major risk factor for cardiovascular disease and accounts for 18% of deaths worldwide. Although the risks associated with hypertension are largely avoidable through effective blood pressure management, nearly half of adults with hypertension have suboptimal blood pressure control. This might be caused in part by variation among patients in their responses to antihypertensive drug treatments. Variability in antihypertensive drug response can be influenced by a range of clinical factors, such as age and ethnicity, and by the level of activity of hormones that regulate blood pressure, such as renin. In the paper under discussion, the authors sought to explain some of the variability in blood pressure response to antihypertensive drugs by identifying single nucleotide polymorphisms, or SNPs, that might influence plasma renin activity. To identify a short list of candidate SNPs, they performed genome-wide association studies, or GWASs, for plasma renin activity in 461 hypertensive patients of European descent and 297 hypertensive patients of African descent. The authors then prioritized the top-ranked SNPs from each of the GWASs by testing for association with blood pressure response to two antihypertensive drugs, a beta blocker and a thiazide diuretic. Because beta blockers inhibit renin release and diuretics stimulate renin release, patients with higher plasma renin activity at baseline would be expected to have a better response to beta blockers and a reduced response to diuretics, compared to patients with lower plasma renin activity, and vice versa. Therefore, the authors hypothesized that SNPs associated with plasma renin activity could be expected to show the same effect. That is, SNPs associated with higher plasma renin activity would be associated with a better response to beta blockers and a reduced response to diuretics. Having prioritized the top GWAS SNPs according to blood pressure response, the authors then assessed each SNP for evidence of functional effects by performing an expression quantitative trait locus, or EQTL, analysis in whole blood from 50 patients of European descent. Lastly, they sought to replicate their original GWAS findings in an independent cohort of 150 hypertensive patients of European descent and 153 hypertensive patients of African descent. The authors found that in patients of European descent, one region achieved genome-wide significance for association with plasma renin activity, that is, with p less than 5 times 10 to the negative 8th, and a further 8 regions met the suggestive level of association of p less than 1 times 10 to the minus 5th. In patients of African descent, no regions achieved genome-wide significance, but 7 regions met the suggestive level of association. Analysis of the SNPs within these regions identified 4 SNPs that were associated with the expected blood pressure response to the beta blocker, atenolol, or the diuretic, hydrochlorothiazide, as shown here. In patients of European descent, 2 SNPs from 2 separate regions on chromosome 16 were associated with higher plasma renin activity and a better response to atenolol, or a reduced response to hydrochlorothiazide. Conversely, in patients of African descent, 2 SNPs were associated with lower plasma renin activity and a reduced response to atenolol, or a better response to hydrochlorothiazide. Next, the authors assessed the 4 SNPs for potential functional effects and then sought to replicate their original GWAS findings in an independent cohort to confirm that the association between the SNP and plasma renin activity was robust. Of the 4 SNPs tested, one SNP, RS3784921, located on chromosome 16 near the genes SNN and TXNDC11, passed these additional selection steps. The results of each of the 4 selection and prioritization steps are shown here. The top panels show the results summarized in the previous slide. The top left panel shows that individuals carrying the G allele for RS3784921 have higher plasma renin activity, and the top right panel shows that these individuals have a reduced systolic blood pressure response to the diuretic, hydrochlorothiazide, as expected. The bottom left panel shows the results of the EQTL analysis. Using RNA sequencing, or RNA-seq, data, the authors showed that individuals homozygous for the G allele had significantly lower expression levels of the nearby gene, TXNDC11, in blood compared with heterozygotes or those homozygous for the T allele. Finally, the lower right panel illustrates that in an independent cohort of hypertensive patients of European descent, RS1802409, a proxy SNP in strong linkage disequilibrium with RS3784921, is associated with plasma renin activity, thereby confirming the original GWAS finding. To summarize, this slide shows the flow of results through the selection and prioritization steps. 4 genomic regions with CHD9, with SNN and TXNDC11, with XIRP2, and with GHR were associated with plasma renin activity and blood pressure response to 2 mechanistically different antihypertensive drugs in patients of either European or African descent. Notably, in patients of European descent, the lead variant in the region near SNN and TXNDC11 was associated with expression of TXNDC11, suggesting a potential functional effect. The association between this region and plasma renin activity was confirmed in an independent cohort, highlighting the robustness of this finding. In conclusion, the authors identified 4 genomic regions associated with plasma renin activity and blood pressure response to beta blockers or diuretics in patients of European or African descent. Genetic variants within these regions may help explain why some hypertensive patients have a greater or lesser response to antihypertensive treatment compared with other patients.